This is episode 80 of the Immunology Podcast, Engineered Biomaterials with Dr. Erika Moore. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology Podcast. We have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Erika Moore from the University of Maryland on the podcast to talk about her research on engineering biomaterial models that harness the regenerative potential of the immune system. We've also got our usual roundup of recent highlights in immunology news coming up, but first...
Explore scientific resources for immunology research at the STEMCELL Technologies Immunology Learning Center. Choose from different research areas and find expert interviews, technical tips, educational webinars, instructional videos, and much more. Visit stemcell.com/immunology-research.
Well, we both have vacations coming up. So yeah, where are you going, Brenda?
I am going to Les Alpes, to the mountains. I'm participating of a fundraising event for the Dutch Cancer Society. And Brenda of last year thought it was a great thing to sign up to. And I'm supposed to bike up the mountain 6 times and not die trying. You know, there's this very famous circuit in the French Alps, which is one of the stages of the Tour de France, and it's called the Alpe d'Huez. And so this event, basically you go and you have to bike, or we can also walk if you want. So I might just take my walking shoes just in case I decide like I cannot bike anymore. And you're supposed to like go up the mountains 6 times. And well, I've also been raising funds. I have a fund me. You know what? You haven't donated to my campaign.
Oh wow, just outing me on the thing.
I know. I will send you a link. I hope to see your contribution, Jason. It's
How do you get down after you go up? Do you have to bike down, or do they take you down in a little van and then let you go back up?
No, I think you bike down because there's no vehicles can be on the road because it's like the only road. So I think the only way to go down is bike down.
How long does it take to bike up?
Few hours.
And you have to do this 6 times.
Well, up to 6 times. We'll see.
But each trip is a couple hours.
Yes. We start at 4:30 a.m. I know. Horrible. Terrible idea. What was I thinking? It's too late now. I paid for the thing. I already collected money. So I'm in too deep now. And I know that you also have very fun plans for holidays.
I'm going on another LARP. I'm going into the woods.
But this is a family edition, right?
It is the family edition with all the kids.
Cute.
But yeah, so that'll be a little bit of a we're going out early because we're helping build the wall for our camp this time. So we bought a trailer, so now we're bringing a bunch of lumber out and power tools and batteries to charge the power tools, and then cords to the very few places that have power at the campground, and doing all the things. So we're starting to pre-pack and get ready and checklists and 3D print things, and also getting in shape. So I've been doing extra working out and such to be ready for the war.
Yeah, because you need to carry like an armor. You said you need to.
Yeah, I do 40,000 steps a day in armor up a hill.
See, not much different to me.
I don't get up at 4:30 in the morning. I get up at 6:30, but
Yeah, it's just one day. I will die.
Yeah,
so if I don't,
4 days.
Well, but it's, you can spread it out. Well, although at your age, I understand that.
Yeah, no, the arthritis sets in pretty quick.
Dear listeners, if we don't make it for the next recording, I died in the mountain and Jason
died in the woods.
Died in the woods. All right, enough chit chat. Let's move on to the important thing.
So one of the ways we could die is through heart failure, and I'm going to tell you about Heart Failure Day. That's a segue.
Go for it. I'll take it.
So in Science Immunology, coming out May 24th, is Heart Failure Promotes Multimorbidity Through Innate Immune Memory. I think this paper is fascinating. Super interesting. Let me get the authors here first. Sorry, so excited. I'm like jumping right in. First author is Yukiteru Nakayama, and the corresponding author is Isesi Komeru. That's first and last. It doesn't have any flags telling me if there's multiple first or last authors on this. So I'm sorry if I missed other people. There's no like number signs or signals for anything. All right. Heart failure, right? Usually you have damage to your heart. It could be from a heart attack. It can be from bad kidneys and overloaded heart pressure, high blood pressure. Your heart stops working as well. And that then takes down other organs, particularly your kidneys, through something called cardiorenal syndrome. Very exciting, right? So, and then worse kidneys make worse heart, and worse heart makes worse kidneys. Then your liver gets involved, and that's hepatocardio syndrome. And then there's hepatorenal syndrome, then you have hepatocardiorenal syndrome, and then you die.
Fun.
Yeah, lots of fun. All right, so they have a heart failure model where they ligate part of one of the arteries of the heart, causes overload, and then you get heart failure, right? And then they do this in mice. But what's crazy is they do this in mice and take their immune cells and give it to another mouse, and that mouse develops heart failure.
All right.
So we're just going to pause there for a second and appreciate that insanity, right?
Yeah.
I'm going to go a little out of order on the paper too and jump to the end where they do a parabiosis experiment. And the so you'd think if it was fully transmissible just by the blood, the parabiosis experiment would be the same in both, but it's not. If you do a bone marrow transplant, using a CD45.2 or .1 into a mouse, then give heart failure and do it, you see transfer. But if you just do parabiosis on its own, it doesn't completely, it doesn't recapitulate, which says there's something, and they already know this. This was a confirmatory experiment, but I like giving a little mystery here. It's not just something in the blood of one mouse causing heart failure in the other mouse. It's what those immune cells experience. So what they find is that, and I'm going to kind of go out of order here, because I actually think I know why they wrote the story the way it did. It reads really well, but for describing it, I'm going to jump. So so when you have heart failure, your immune cells for the initial injury seem to be preconditioned, and they are preconditioned, and they see this through the chromosome accessibility. It suppresses TGF beta, and what they see before they figure that out is that causes monocytes to be less likely to become tissue-resident macrophages. And it's a separate population. So they do a bunch of lineage tracing and a bunch of immune cell barcoding to track all of this. Or they'll barcode the cells and transfer them over and see where they go. And what they see is that different cells end up doing different things, right? But the ability of these heart failure conditioned cells, these monocytes in particular, to become tissue-resident macrophages is diminished. The rest of the population is fine, but the ability to do this is diminished. And these tissue-resident macrophages are fundamentally important for preventing heart failure or for not exacerbating it. So when you don't have them, you have exacerbation of the disease in other mice. And then if you do a renal injury with these immune cells put in from a transplant, renal injury is worse. So having this lack of tissue-resident macrophage capacity makes renal injury worse, which is that cardiorenal syndrome. Now, what the heck is going on here? Well, they actually get down to isolate that it is sympathetic nerve fibers in the bone marrow. Like, all right, this paper's just wild, right? Now I'm talking about the nerves. Sympathetic nerve fibers in the bone marrow that release TGF beta and condition. And so what's happening is that, and they can't quite figure it all out, which is why it's probably science, immunology, not science. But that injury in the host changes those immune cells, which is also where the parabiotic thing doesn't transmit. That injury in those nerve cells through TGF signaling is conditioned to be then down-regulated on the TGF axis. And those cells are permanently fate altered to be unable to make as many tissue-resident macrophages. And that leads to the exacerbating injury and heart failure.
That's pretty insane.
Right?
So sorry, but the neurons they get like it's the heart failure, then it goes to the neurons through the neurons to the bone marrow.
No. So during an injury response, like during
Uh-huh.
a heart failure injury, there's an immune response, right?
Yes.
It's bad. Your immune system's going, and the sympathetic nerve fibers in your bone marrow signal through TGF beta to make your monocytes become tissue resident macrophages and fix that stuff. If you take that out and put it in other mice, if you take those cells out though, and the heart failure turns off the TGF beta. And so they're unable to make macrophages. And so if you take that initial injury preconditions you to get more injury. So if you take the preconditioned immune cells and put them in a new mouse and give it heart failure, it gets worse heart failure. If you give it kidney injury, it gets worse kidney injury than normal immune cells. So the heart failure is a chronic condition, right? So you have injury. It's not the first episode that gets you. It's the fact that you get it and it gets worse over time. So it makes itself worse. And this is how
Okay.
it makes itself worse. In addition, you get spontaneous worsening of heart. If you take these mice and let them get old, 6 months out, you get worse heart failure. They get spontaneous low-level heart failure. If you take the immune cell from one to the other, right?
Is that the motivation for this? Is studying why heart failure gets worse
Right. Like
each
it's one
time?
episode of heart failure up, but once you have it, you can't get rid of it. You're just trying to hold it off from getting worse and boxing your kidneys, and your kidneys get worse, which we know that makes your heart failure worse because the worse kidneys causes high blood pressure, and high blood pressure makes your heart failure worse, and high blood pressure makes your kidney failure worse, and then they make each other worse, and then it's just the spiral of death.
And what motivated them to do the bone marrow transplant? What was this?
They were trying to see if the immune compartment was involved at all. So if you just take like I give it heart failure and move it over, does the immune cells from the first injury transmit? And the answer is yes. And preconditions a new mouse if you injure it to get worse disease.
All right.
Or if you wait long enough, right? Heart failure is usually in older people, so you don't see this phenomena that much. You don't have like a young person get heart failure and then wait. They're going to get, they're not going to get spontaneous. That's why the transfer is only an experimental thing. But if you take the immune cell, put it in a young mouse, and wait, they do develop spontaneous heart failure when they're older.
I see. Wow. Do I have a segue for this? Not really. But it's another EBV story. So Epstein-Barr virus is quite a character, isn't it? It's just nowadays it's just popping up everywhere. And I think we already knew it can give you leukemia. We are, more recently understanding that apparently it also gives you multiple sclerosis. So this paper is titled A Metabolic Dependency of EBV Can Be Targeted to Hinder B Cell Transformation. And it was published in Science. First authors Bojana Müller Dubrovic, I guess, and Jessica Jäger from the lab of Christoph Hess at the University of Basel in Switzerland. And I thought it was very interesting because, well, so we know that EBV has the ability to transform, particularly B cells, and render them cancerous, basically. I mean, there are different levels of transformation. You can also have like lymphoproliferative disorders. So there's different degrees to which EBV will generate these problems. But basically, the following disinfection, particularly in older people, this can happen that the virus engages pathways in the host that mediate cell proliferation and transform the cells. And one of the things they were looking into is trying to understand which are mechanisms that have to do with metabolism of these cells, and how does EBV kind of drive some of this transformation. Um, so what they did was to transform. So they have naive B cells um, in culture, and they infect them with EBV, and they look at how, especially the initial steps in the transformation of these B cells. And this is trying to understand that transition between a normal cell and what are the initial steps in the transformation. So one of the characters in this study is EVNA2, which is basically this viral derived protein which acts as a master regulator that orchestrates a lot of the cell activation and cell cycle entry of EBV infected cells, and its function is required for the transforming effects of EBV infection. So, what they do when they look into the early stages of infection of B cells with EBV, they try to find, they do a metabolic analysis, compared, uh, EBV infected cells with normal cells, and there's one particular metabolite that stands out, which is quinolinate, and this is a metabolite of tryptophan. And it's part of a pathway that can be used for NAD de novo biosynthesis. And they found that interesting. NAD, of course, is a very versatile metabolite in cell metabolism. It's required for redox activity throughout the cell, but particularly as an electron as an acceptor of electrons during cell respiration in mitochondria. Uh, the production of these upstream of quinolinate, so downstream of tryptophan, is this IDO1 enzyme, which catalyzes the first and rate limiting step of the tryptophan catabolism. And what they notice is an upregulation of IDO1 in these cells during the early stages of infection. So basically, they see an acceleration of this, what is known as the kynurenine pathway, which includes a consumption of tryptophan early post-infection and a temporary increase of the catabolites of this reaction, and a support of NAD biosynthesis. And this is very important because it is really the expression of IDO1 during this early part of the infection really is associated, they find is really associated with the early infection and this initiation of NAD production in infected cells. So they look a little bit to try to understand why is IDO1 being upregulated upon EBV infection. They see that IDO1 is an interferon responsive gene, and the upregulation of IDO1 is dependent on this EVNA2 viral protein, and they use they find with some cheap some immunoprecipitation, they see that the EVNA2 interacts with a factor called EBF1, and this allows it to interact with the DNA of IDO1 to induce the expression of this enzyme. And basically, this the increase in this pathway results in an increased NAD biosynthesis, which is important for the induction of mitochondrial respiration. And I guess this is a way in which this transforming cells can fulfill the energetic requirements of this viral infection. But I think what's interesting, this only really applies to the early stages of infection. They don't see this upregulation of IDO and this NAD biosynthesis is most critical, like in the first 4 or 5 days. But if they interfere with this pathway later on, it doesn't seem to matter as much. It's like once this transformation is ongoing, then there's a more of a more the requirements change more to glycolysis, and so therefore it's not so critical to have this mitochondrial respiration going. And what I thought was very interesting is that they try to think what can we do with this information. So we know that IDO1 and tryptophan catabolism is important, and NAD production is important for these early stages of for generating a transformed B cell. So if you interfere with these B cells are not transformed, you don't have lymphoproliferation. And this is particularly important in a subset of patients in which they are EBV negative, which is fairly rare, right? Most of us are EBV positive. It's such a widespread virus. But there's a situation in which people receive transplants from EBV seropositive people, but the recipient is they're themselves negative. So they really are subjected to the risk of acquiring EBV during the worst time when they are immunosuppressed because of the transplant. So the idea is that they wanted to, so they look into these patients and they show that again, there is an upregulation in the very early times in those patients that ended up developing a lymphoproliferative disorder because of the an EBV infection. They see this presence of circulating EVNA2 and IDO1 positive B cells and activation of this kynurenine pathway right after the transplantation, suggesting kind of a causation and that kind of part of the development of this proliferation. So they suggest that by there's inhibitors of IDO1, and they suggest that by trying to treat patients right after with in this critical window with this inhibitor might reduce the chance of developing transformed B cells due to EBV infection. So I thought it was very, very interesting.
How to keep it from popping back up. Yeah, no, it's a major problem and a big boogeyman of donor screening, transplant screening, everything else, but can be hard to detect unless you have, what does an IgG positive mean, right? Like, oh, you had it at some point, right?
Yeah.
Well, so did everyone. If you have IgM, great. You shouldn't donate, but that's it, right?
Well, but if you can do something to like
shut it off.
Shut it off. So they show in them some mouse, like humanized mouse models, that this might help. So, but it doesn't help once the transformation is established. It doesn't help, like transformed cells don't respond to this. It has to be this critical transformation process that gets affected.
All right. So next up, I don't have EBV. I don't have anything cool like that. I just have a gut microbiota paper. So in Cell, and making its rounds on the microbiome world of LinkedIn and Twitter, we have gut bacteria convert glucocorticoids into progesterones in the presence of hydrogen gas. First author, Megan D. McCurry. Last author, Sloan Devlin. Coming out in Cell in June 6th. Okay, I'm going to cut to the chase. So, corticoids, right? Standard stress hormone that can be found in the bile. We know the bacteria can metabolize these sometimes, but understanding this in detail has been tough because getting enough bile from a person to be able to do the studies is hard. And so the first thing this paper does is they put together a new mechanism using mass spec and other fancy stuff. So they use a combination of ultra high performance liquid chromatography, atmospheric pressure chemical ionization, an Orbi-trap mass analyzer, single ion monitoring, and tandem mass spec. So UHPLC MSMS, they are able to get down to 100 microliter samples of bile and get limits of detection 2 or 3 better fold lower than with GCMS that was previously done. So lets them isolate other isomers and other things that were hard to separate before from biological samples. So that's the first key thing to go with. All right, so knowing that, going to cut to the chase here. So 21-deheroxylation has been known in eukaryotes, is not observed in karyotes. It's always been thought to be in the microbiome, but figuring out what bugs does it is hard, but we know stool can do it. And that converts corticosteroids into progestin, sex hormones. Okay. So step one is that they show that, but it's not very common. But what they find is that in pregnant mice, it is more common. So the stool of pregnant mice does this much better than the stool of non-pregnant mice, especially late-term pregnancy. Okay, well, that's interesting. But then they find that these metabolites are enriched in the feces of pregnant human donors. Well, that's really interesting. And then they identify the species that do this. Gordonobacteria and Agaricella species do this, and then they isolate the gene clusters involved that do this. But that's not enough, right? That's already ridiculous. because that's already hard work to identify the clusters and get it out, and they have to clone it and knock it out and induce it and put it in something else and show that it works. All right, so they can show that like this other bacteria with the cluster works. But they also find that, some of these Egerthella species don't do it very well without the presence of E. coli, Nissen, or other bacteria. It's co-cultures seem to work. And by the way, it took them a while to isolate the individual bacteria. They had to do these enrichment studies with arginine to make these bacteria grow up more, and then do and then they did single colony picking and didn't get anything, so they did quarters of plate. So they said a quarter of a plate that did it, then did single colony picking of that. So it took work to find the bug. But then they find that that H2 producing bacteria, because they find that the higher the more negative the redox potential of the milieu, the better this enzyme works. Well, they figure out two things. One, they figure out it's a molybdenum, cannot pronounce that heavy metal based enzyme that uses it. But they also figure out that it uses H2 likely as a substrate, and that is made by other bacteria. So other bacteria that make H2 make this more potentially useful. They grow it up in H2 environments, and it's better. If they do an N2 environment, it's not. So they isolate these bacteria, establish that they're there, establish that they convert stress hormones to sex hormones, establish the enzymes that do it, establish that other bacteria are needed for this reaction to occur in human, and establish that in pregnant people, there's more of these bacteria. And guess what? More progesterones protect you from depression in late pregnancy and postpartum. So your body is reacting to being pregnant by having a microbiome shift that results in different metabolites occurring to make you less depressed during pregnancy.
Is that the baby glow that everyone talks about?
I don't know, but like literally your microbiome is being sculpted by pregnancy to shift its metabolite production to augment the beneficial outcomes of pregnancy. And of course, this works by FMT. I'll just add that in now.
Wow.
And germ-free association studies. There you go. The microbiome is a symbiote.
I'm not sure how I feel about this mind control by our microbiome, but well, fascinating. Fascinating. Well, just to quickly finish our conversation today, another fascinating topic for me that I don't understand enough about and I should probably get to know better is the effect of the circadian rhythms on our immune system. So this paper is called Circadian Control of Tumor Immunosuppression Affects Efficacy of Immune Checkpoint Blockade. First author, Bridget Fortin, from corresponding author Selma Masri at the University of California, Irvine. And the paper kind of starts with our understanding that there are rhythmic changes in our immune system. That's something that has already been described. And there are time of day dependent trafficking of immune cells, composition of certain parts, and we know that this affects like things like cancer therapeutics. There's evidence that it's not the same to administer chemotherapy in the morning or in the evening. That immune checkpoint studies show that afternoon dosing is worse, has worse outcomes. And so I think we start to understand like this is pretty complex, but they're trying to kind of approach this and understand what is the effect of the circadian rhythms on our immune system and cancer. So what they do, they have this genetic model of colorectal cancer in mice, in which they knock out this APC tumor suppressor gene, which I think is very common for this cancer model, specifically in intestine epithelium, and also they knock out this member of the circadian clock is BMAL1, and specifically in the intestine epithelium, so just there. And they do all the very fancy single cell RNA-seq, and they show that this disruption of circadian rhythm by the epithelium of the gut changes the composition of the cells in there. Um, so basically, what they see is an increase in inflammation, uh, increased proportion of CD8 cells, more neutrophil recruitment. And, long story short, they do a lot of characterization. I think, long story short, they see that this change, mostly, I think the most change they see is neutrophil recruitment and activation of T cells. Um, and this is due to the effect of wind signaling that is also affected by the circadian clock. So this is how they connect these two things. But they they find that the myeloid derived suppressor cells that are, in a situation, in a model of cancer, are preventing immune response and response to, for example, checkpoint inhibition. They are also fluctuating due to the circadian rhythm. Um, so in a model of colorectal cancer, they see that the immunosuppression that they observe is mostly likely mediated through this clock-dependent control of myeloid-derived suppressor cells, um, and through the Wnt response. So they have this inflammatory response that recruits these cells to the area. And this, in fact, has an effect on checkpoint inhibition treatment that they do in these mice. And they show that by having different, they do PD-L1 treatment, and they show that if they have it at different times, so the efficacy of the PD-1 L1 therapy is influenced by the timing of administration. So they have this times. Of course, the mice and humans have different times. So what is it? A rest for a mouse is different for a human, but they take that into consideration. And basically, the conclusion that they arrive is that delivering PD-L1 treatment when these myeloid-derived suppressor cells are the most abundant, which in this case is the early active phase, so what they call, I think, Zeitgeist 1 or 4, no, it's like 4 hours after starting the day, it enhances the efficacy of the therapy. And this is due to this cyclic uh, um, activation of the immune cells through this Wnt signaling in the intestine. So I think really shows that we have to be so careful about these things. I find it, yeah, mind blowing.
I wonder if the Wnt signaling cycle with the rhythm is based on natural regenerative rhythms that will go through, like regenerate while you're asleep type of thing. Right, because you tend to wound heal more when you're at rest, and so that's driven by Wnt in part.
Well, but if it's true that so Wnt is affected by the activity of this circadian clock associated protein, so yes.
Yeah, I'm wondering that's like where the evolution of it comes from, is from the
Oh, I
approach,
see.
right? Like why is Wnt regulated by your circadian clock? So you heal when you're sleeping.
Yeah, that makes sense. That makes sense.
I may have spent some time with APC mice and cancer and Wnt.
Yeah.
And beta-catenin.
Yeah.
All right. Well, we're going to be speaking with Dr. Erika Moore at the University of Maryland in just a moment. But before we get to that, are you using macrophages for your research? With Immunocold SF macrophage medium from STEMCELL Technologies, you can differentiate human monocytes into macrophages. The medium and protocols available can be used to differentiate the human monocytes into M1 or M2A macrophages in a 6 or 8 day culture period. Learn more at www.stemcell.com/macrophage medium.
So now for our second part of the show, our interview, we are very happy to have with us Dr. Erika Moore. She's an assistant professor at the University of Maryland, and she's going to be talking to us about her research on biomaterials and engineering biomaterials to study, among other things, immune cells and how they work. We're very excited to have you on the podcast. Dr. Moore, thank you for joining us.
Thank you all for having me. I'm so excited to share some of our work with you.
Well, thank you for being here. All right. So tissue engineering and matrices. Could you kind of, for people who are maybe coming at this from an immune cell perspective, describe kind of the playground and tool set you work in and how the engineering gets to the immune cells?
Yeah, that's a great question. I think I like to start in context, right? So cells often just don't exist by themselves. They interact with lots of other cells, proteins, et cetera. And so where my group started is really understanding the tissue microenvironment that cells exist in. In particular, I focus on the innate immune system. I really love monocytes and macrophage immune cells. And so we wanted to study, okay, how do signals that macrophages and monocytes receive from their tissue microenvironment change, affect, or direct the cell function, right? And so we started by using these cool materials called biomaterials. And for those who are unfamiliar, biomaterials are honestly any material that's created that uses or interacts with living beings, right? So it could be something like the contact on my eye, that's a biomaterial. Any person who knows someone who has an implant, those are biomaterials in their bodies. And in my group, we actually use synthetic polymers called polyethylene glycol to create different tissue microenvironments. And we can control the microenvironment. We control basically what the cell sees, and we study how that affects cell function. So we look at a lot of different functions of these innate immune cells, but we do it all within this biomaterial tissue construct that we create in the lab.
So let's talk about why we should be paying more attention to this third dimensionality. Because what's wrong? What's wrong with my, my plates, my 612 plates, my 9612 plates? What exactly are the interactions that we are missing when we're talking about immune cells? In, for example, I know that you have some kind of biogel structures, and you use that to study in lupus and other diseases. So why do we need to care about this?
Yeah, this is so great. I think there are so many different models that we can use in biomedical science. Some people use animal models, some people, like you said, use these tissue well plates. All of these are contributing factors to helping us understand fundamental immunological behavior or profile. Where we like to come in with our tissue constructs is they allow us to study these cells in a environment that mimics some aspects that we see in vivo, like in a tissue environment. And what's so great about these is we can account for differences that are seen. Maybe someone develops a disease, maybe another person doesn't, and we can isolate both of their cells from those human patients and study them, study how those cells respond in our tissue construct. So what's missing right now is having these kind of intermediary model systems that still mimic certain aspects of the tissue microenvironment, but allow us to still relate them back to human beings. So, for example, one of the model systems that we created in our group really looks at how aging of macrophages affects their function, and specifically their ability to support vascular repair or vasculogenesis. And what we saw is it's really hard to do that in aged mice, because aged mice are really variable. They have a lot of fatty depots, and they can sometimes just randomly die on you, right? And then also, it's really hard to study that in a 2D environment because we wanted to understand how these cell-cell interactions, both the soluble secretion as well as the physical cell, touching the juxtacrine interactions would affect the cell function. And so in order to do that and still have it relate to humans, we wanted to get our human cells and we wanted to put them in kind of our tissue microenvironment, these synthetic polymers that we're able to mimic. The other example I'll give you just really quickly is we also have this whole other project that looks at how integrin ligands that are present on the surface of certain extracellular matrix proteins actually directs macrophage function. And that would be really hard to do in a really complex environment like an in vivo tissue. And it's really also hard to do in 2D, right? Because we don't have great control over the proteins that are absorbed onto the surface when we culture those cells in 2D. So when we move into a tissue microenvironment, we actually have total control over what the extracellular matrix proteins can look like. And thus we can really pinpoint, okay, how does this tissue signal to these cells that are in this matrix, and how does that affect their function? So those are two examples of the gaps we try to fill in my group.
So how do you make your 3D biomaterials, and how close are they to what's in me?
Yeah, that's a great question. So we make our 3D materials using actually a pretty common polymer, as I referenced earlier, polyethylene glycol. And we do a host of different chemical reactions to functionalize them to make them a little bit more similar to our tissue microenvironment. So we add in extracellular matrix adhesive peptides. For those of you who are familiar, maybe you've heard of RGD. There are actually a host of a lot of different cellular adhesive peptides that we can add into our system to mimic certain aspects of the tissue microenvironment. And then we also add in peptides that are sensitive to matrix metalloprotease. These are secreted proteases from cells that actually degrade our tissue. And so if you think about comparing them to, your actual native tissue, we're still a little bit further away. We have two main components, right? The cell adhesion and the cell mediated degradation, but we don't, necessarily have some of all of the elements that you might see in a tissue environment. What's still cool about our system, though, is we're still able to kind of take this reductionist approach and control which variables we want to study, such as stiffness, different mechanical cues, and basically isolate or combine them all to understand mechanisms of action in our monocyte and macrophage immune cells.
So I saw many of your work is looking into particularly macrophages, monocytes, and microvessel structure. And so, would you like to maybe share with us some of the key insights that you have learned related to how does, for example, integrins or peptides that are derived integrins affect macrophage polarization? How does this relate to, tissue aging and response? You have work done on responsive metformin, on the microcell structure in lupus. So, what are the key insights that you would like to kind of share with our audience?
Oh yeah, that's a really great question. I think the first key insight, which is kind of the first major project that we spun out in the group, is the fact that macrophage function can be attenuated through targeting extracellular matrix ligands. And so specifically, some of our earlier work saw that if we target certain integrin receptors like alpha 2 beta 1, we can actually block or inhibit macrophages from becoming really pro-inflammatory. So we can reduce upregulation of inducible nitric oxide synthase, INOS. We can also reduce expression and secretion of tumor necrosis factor alpha, TNF alpha. We can also reduce expression of, a host of other cytokines. We assess both gene and, protein secretion responses. And all of that can come from just targeting a specific integrin receptor. And so this was actually pretty cool, right? Because most of the time when we think about macrophage attenuation, we think about soluble mediators like delivering, really potent anti-inflammatory or pro-inflammatory mediators. But in this case, we saw that if we just actually target the integrin receptors, we can preserve viability, right? So the cell's still alive, but it responds totally differently. And so right now we're working on building an entire atlas that will allow us to categorize all of these different integrin receptors and how they can change and alter macrophage function. So that was one of the first major takeaways. The integrin or the extracellular matrix that the cells exist in actually really matters for determining their function. The other aspect is the one you referenced about aging. And this actually was kind of a project that was born out of some of my curiosity. As we age, we know kind of we heal and repair less efficiently. So we really were interested in understanding how macrophages that were isolated from donors who were 65 to 72 versus donors who were about 21 to 35 really behave differently. And what we saw is that, macrophages that were isolated from older donors actually support a reduced form of vascular development or vasculogenesis. And this was really evident in the fact that there were pretty substantial differences in what we quantified in vascular plexus formation in our assays, in our 3D structures. And so this was really cool because it allowed us to isolate the fact that, okay, age plays a really important factor in our ability to repair vascular plexus. But then we tried to rescue the function by using our platform to screen and understand the response to different drugs. So we used an anti-aging therapeutic, metformin, and we basically saw that if we treat aged macrophages with metformin, we could repair or restore some of their function, largely through targeting monocyte chemoattractant protein 1, MCP1 upregulation, that upregulated a host of other pro-angiogenic cytokines like VEGF. And then the last project that I'll briefly touch base on is our project that's ongoing with lupus, where we've really seen that, in lupus, clinically, there are some alterations that are seen in microvessel function and formation. And so in our group, we've been able to isolate cells from lupus patients and understand how those cells we're seeing differences in microvessel organization based on disease versus a healthy control donor. And so it's really exciting because it's introducing this preclinical model for lupus and understanding the interactions between monocytes and endothelial cells as the vascular plexus forms. So those are some of the main takeaways. I know that was a lot. I'll pause and see if there are any other questions you want me to dive deeper in on.
I actually wanted to go back to PEG for a second. I think about this from pharmaceutical land, where PEG is a very common excipient, but also highly antigenic to the place that it often gets an anaphylaxis warning if you inject it or administer it any way other than swallowing it. And so I'm wondering, do you guys know if, especially with immune cells, if a PEG matrix is immunogenic in a way different from host or beneficial because you need it because everything's artificial and that's a feature? It could be a feature or a bug. I don't know.
Yeah, that's a really great question because we're definitely aware of the response to PEG, especially clinically. And I mean, you see PEG everywhere. Right now, we also do studies in other materials, right? So we do studies in collagen-based gels and fibrin gels. And thankfully, we've seen our trends and responses of the macrophages be conserved, right? So at this stage in an in vitro setting, we haven't seen major differences in response to the macrophages to our PEG hydrogel system, especially compared to other collagen or ECM-derived biomaterials. But it's definitely something we're highly aware of.
Right, because especially if you want to then translate it to more clinical, if you ever wanted to, I don't know, bring in some cells using that PEG, that's definitely not going to work, in a patient. I just want to say, you see, Jason, I hope you're excited about all these anti-aging research being done, by the young people on your behalf.
It just depends on if it gets done in time now at this point. I
It might be too late for you. I'm sorry, Erika. This is how we run. This is how we roll with
maybe a little bit late, but we'll see. It depends on how much reversal you can do.
Well, we'll get all the M2 macrophages we can muster for you.
Yeah.
Erika, if I may switch gears a little bit, I think it's very nice when we have also a guest in our show that has, some extracurricular interests. And I know from your profile and from the work that you do that you also have a very strong interest in kind of equity in bringing your research to a more to benefit more people. You also have a very interested side hustle, if I may call it, at helping with financial literacy of students. So I would just like you to maybe give us a little taste of the things that motivate you besides your work and your lab and your students, because I think it's so interesting to get a more roundup vision of the researchers that we invite on the show. So maybe would you like to tell us a little bit about that and the motivation behind it?
Yeah, absolutely. I'd love to speak to some of my motivations. I think from the outside looking in, it seems so random. But for me, a lot of my motivations come from a collective of my own experiences, as well as the experiences that I've been fortunate enough to be around with other people. And so I think, I'll start with financial literacy. When I was in undergrad and grad school, I experienced a lot of financial stress. I took out a lot of student loans to go to school. And I just remember thinking, it would be such a privilege to be able to just focus on my classes and not worry about the finances of paying for my degree. But that wasn't a privilege that I had at the time. And so I started to learn a lot and educate myself. I interviewed a lot of people, and I became like best friends with the financial aid office. And so from there, I realized that there is actually a dearth in understanding or communication around financial literacy or personal finances for a lot of students, like especially those who are from, just different backgrounds. Like maybe you're the first person in your family to go to college, or maybe you're the first person in your family to just go to a private institution versus a public institution, right? And so the foundation of financial literacy and personal finance I found was just really absent. And beyond that, it actually creates a huge barrier to access. And so that's why I started working on financial literacy. And then for the other equity, considerations that I do in my group, it's very much so motivated by my own interests and the interests of my students and my trainees. I think a lot of us are kind of like coming to the table. We're really excited to be biomedical engineers and we want to contribute to this growing community. And so we're like, how can we become more equitable? How can we increase access? How can we ensure other people who are interested in this work can also join us on this journey? So I think it's largely just based on, a collective of experiences and interests, and really how much can I fit into one day? That's mainly the main question.
So just to wrap up on that topic. So one of the things when it comes to really addressing this, for example, in the lab, like really improving our scientific approach to our studies, I think one of the points you have made in the past is about the origin of cells or understanding which are the models that we're working. I think in general, biomedicine has a very strong kind of white Caucasian bias when it comes to the patients, the cells. So where do you think we stand in that sense? If I assume you have an opinion or maybe a suggestion on policy, where do you think we should be working on? Not from a pragmatic scientific point of view. I think let's assume this was an honest mistake, and then where do we go from there?
Yeah, I think there are so many places to go from there. I think a lot of people are intimidated because they're like, well, we can admit something was not great, but what's the next step? And I always start simply by saying, if people are doing studies, report the cells that you're using, report de-identified information that can be shared. Sometimes people have IRBs that inhibit them from sharing other things. But like, for example, even if you order cells from some of the largest manufacturers in the world, they can give you coded de-identified information such as age, such as demographic descriptors, and sharing that could also help us to basically build a patchwork where we can basically kind of connect and compare our data, right? So if I get this response in B cells, well, and now I know someone else is using this kind of cell, so it makes sense why we're not getting the same response, right? Or why it is the same response, and that to help us connect even further. So I think reporting, communicating intention, and also considering which disease you're studying and who's affected by that disease. So, for example, we study lupus, and lupus is a very gendered and biologically sex affected disease. And so 90% of the people with lupus are women, which means that it wouldn't really make a lot of sense to do all of our studies in using male cells, right? And so we have to acknowledge that in the work that we do, and the diseases that we're trying to model and understand. The same thing goes with aging, right? Like aging looks very different based on biological sex, and it really has to be considered in these models that we create and try to communicate with others.
So, what about the bias, though, in clinical trial populations and the barriers to overcome that? Because I worry, we don't have equanimity in a lot of ways, but then also when you try to go get it, you're told to go take a hike. I don't want to be involved, to be real blunt about it. And so then you can't get what you're looking for because the other side has to consent, and they don't.
Yeah, I think it's a larger problem. You're right. There's a lot of medical mistrust and hesitancy, which I think is well deserved, right? Like you can't try to fix a massive problem that's been ongoing for I would say centuries with just kind of one size fits all, quick like just volunteer approaches, right? So it does take collaborations with a lot of social scientists, anthropologists, and we actually even work with a ton of community centers that allow us to partner with and communicate with vested, people who are vested in our studies. So whether that's kind of understanding and communicating what we do when we get their samples, that's largely on us and the teams that we build, right? And so then that helps other people become less hesitant, hopefully. But it's definitely a much more challenging problem to solve. I understand it and appreciate it, and I'm doing my best to try to work on it. And I think we actually are working on it by communicating, right? Like this is one form of communication, sharing what goes on in the lab, right? Some people aren't familiar with that. And so appreciating that we all come from different perspectives and have to do our best to kind of bridge the gap in communicating and understanding our own biases so that other people feel comfortable participating and helping us close some of these gaps.
I think definitely very important to address the gaps. So I think we're reaching the end of our conversation. It's been very, very interesting. We'd like to kind of quiz our guests a bit at the end. And I think probably one of the favorite questions to ask is if you were not, a successful assistant professor at the University of Maryland, if you're not, we're not a scientist, which I think probably this has been going on for a long, long time, but what would you be? So what would be your alternative universe option?
Yeah, I think I would be a writer. I'd actually love to write books or maybe a book editor. I'm not sure, but I love writing and reading. So and fiction too. Like so this would be all fiction, nothing science related.
Very nice, very nice. Any book you're reading lately that you might want to recommend our audience?
Yeah, I actually just finished. This is actually nonfiction. Hidden Potential by Adam Grant was excellent. And if any scientists are listening and they are kind of like, am I cut out? How do I make it? In this kind of competitive, sometimes ostracizing environment, I highly recommend it because Adam does a great job of communicating different ways to grow your character and strengthen yourself so that way you can survive and actually achieve all of your potential.
That's very nice. I mean, Adam Grant, no, for those is a very well known author in this field, right? Of
Yes.
kind of working, how to work better, how to. Very nice. And this is I had not heard about this particular book from Adam Grant.
Yes. It's his latest one. I highly recommend it.
Excellent. All right.
Well, I think that covers it. Well, thank you for being on. How do people find you on the socials and such?
Yes, I'm on Twitter at Dr. Erika Moore. I'm on Instagram at Erika Moore Taylor, and I'm on LinkedIn just at Dr. Erika Moore. So if you find me on any of those, you can also just feel free to Google my name, Erika Moore, Moore, and hopefully I show up.
Wonderful. It was great having you. Thank you so much for sharing this conversation with us. So Dr. Erika Moore at the University of Maryland today joining us at the Immunology Podcast.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on X at @immunopodcast or via email info@immunologypodcast.com with feedback or to suggest guests. See you next time.